So small. Share screen. Okay. Good morning, everybody. I'm gonna put my chat up.
Good morning.
Good morning. Okay. Like all the different things. What am I doing? Okay, attendance first. If this goes away. All right. So you can check in with attendance whenever you get a chance. Is it cold in here today, or is it just me? Oh, 67.
It was 168 and it just went down.
It's warmer than the lab, I'll tell you that.
Yeah.
What's more than the blood?
It's warmer in here than it is in the lab, but it's not saying very much.
Oh yeah, yeah. It's cold.
Is the AC in the lab working or is it set?
It's not, no, it's like set to 74. I don't know. I don't know what to tell y'all. All right. Wish I would have brought my jacket. It's all right. Yep. There we go. And I'm just going to monitor the chat on my screen. Got a few minutes. So y'all watching the hurricane?
My dad says that's gonna keep going. It's gonna keep going, like, but it's just gonna keep going west of turn.
It's always unpredictable.
I keep telling.
Well, it's unpredictable because we were like, oh, we're tracked for this one, we're tracked for that one, and we didn't get hit. But
I still say,
if they don't track us, I'm gonna think we're more likely to get hit.
yeah, I know for a fact the school's gonna keep it on high, like, they know just on the off chance hurricane does happen and they don't cancel classes, they're gonna jump on, they're gonna sue their ass for.
I know we're gonna be in trouble, so. I think Friday. Oh, they changed it to Saturday? Saturday morning. They just updated the plot, and it is closer to Lake Charles now. Oh, poor Lake Charles. Jeez.
Poor Lake Charles.
Oh, man. Just bust us over there and work. Help each other. Yeah, that would be a good thing. That would be a good thing. What time we got? I don't even know. So don't forget to check in on attendance. 2 minutes. We got 2 minutes.
Wow, we are real slow on responses today, aren't we?
Slow on responses? Yeah, I know. We're slow on responses. Only 29 responses. It happens. We're going to roll with it. Y'all want to hear a joke?
Knock it out.
Knock us out. I'm telling you it's a joke on the front end. Wait. I'm not going to mess up the punchline on this one. All right, you ready? Where do you take someone who has been injured in a peekaboo accident?
I don't know where.
The ICU. Thanks.
Sound very proud of that one.
I kind of do.
Did your son love that one?
He didn't. He didn't even think it was funny at all. At least with y'all laugh. But he says none of my jokes are funny, and then he laughs at like when I tell him to go clean his room. I'm just like, can't win. I just can't win. It's okay. Yeah. Okay, so everybody should have checked in on attendance, right? Yeah, go ahead. I'm gonna leave it up for a little bit because I'm not doing a clicker question yet. So you had to watch 7.1 before you came to class today. And
Yeah, I did that.
further, oh, I keep going the wrong way. You know, my mouse in my office scrolls one way and the mouse here scrolls a different way. And it's really messing me up. Okay, let me just go down. All right. So we started talking about enzymes, right? And kind of their general function and things like that. So we are going to pick up there. You're actually going to get a lecture today. I don't know if that's a good thing or a bad thing for y'all. Let's see. I want to do this one. No, you. I want full screen. There we go. Then at least I can write on it. Okay. All right. And I know y'all are waiting on grades. I'm working on grading them. I'm going to try and get them graded this weekend so you can have your results, because we only have 2 chapters on this next exam, right? We only have 6 and 7 and 8. Is it what? Yeah. Yep. Just because of the topics, it just works out really well to group these together and then to have the next chapters grouped together just because of the topics. And so you'll have a shorter amount of content on the next exam. So that should be kind of nice. That should give you a little bit of a break, right?
So what you're saying, this big test is 2 weeks?
Yeah, it's about 2 weeks.
Okay.
It's about 2 weeks.
Fair enough.
So the key to studying for this is a little bit all the time, right? A little bit all the time. All right, so enzyme structure and function. So you have to remember that all of these enzymes are functioning inside of our body at physiological conditions. What are physiological conditions? What are some examples of physiological conditions? pH 7. What else?
98 degrees Fahrenheit.
98.7 degrees Fahrenheit, but you know that's the F word, right?
Yes, I know. We're
That's what I call it, the F word.
Or like 20 something degrees Celsius.
Yeah, well, 37. 25 is room temperature, 37 Celsius. Okay, what else? So molarity, and I'm going to say molarity of lots of substances, not just water, but I'm going to say physiological concentrations. I'm just going to say of everything, right? Because you can't get crazy concentrated. You can't get in either direction, right? It has to kind of be one or the other. Don't forget to, yeah. These are all physiological conditions, right? How about pressure? Can we change pressure inside the cell? It has to be constant, right? So you're basically at 1 atm, basically. So all of these things are things that can't change. So when you were in general chemistry, how did you change reaction kinetics? You heat it, right? You change the pressure. You change the concentration. Can we do any of that? Not really. We really can't. We're stuck. We're stuck in this physiological system. So we use enzymes instead. So enzymes are going to increase our rate of reaction, but we're not going to mess with all this stuff that we wrote up here. We can't mess with it. So they're going to lower the activation energy. How does that happen in order to increase our rate of a reaction? Well, we're going to stabilize our transition states. Do you remember on your reaction diagram, right? If you had something here and something here, we had to go through a hill in order to get there. Remember that? Okay, so that transition state is something that an enzyme can stabilize. We're going to provide an alternative path for product formation. So we might not want to go to this particular intermediate form. If we could go to a different intermediate that has a lower energy, what's going to happen to our rate? It's going to go faster, right? Because it'll be a smaller hill to have to climb. That's the point. So a lot of times this is an alternate intermediate. And a lot of times that alternate intermediate actually participates in a covalent bond with the enzyme. And we'll look at some examples of that in this section and in the next section. And they're going to reduce entropy. They reduce entropy by orienting our substrates appropriately. So how do you know a chemical reaction is going to happen? If you have substrate A and substrate B, they have to be oriented in the right position in order to create a new product, right? What if they're oriented strangely? Is it going to go? It has to be a productive collision. Y'all are a little bit asleep today, huh? I feel like I'm making a recording and not teaching in class.
I'm trying to turn down. I'm sleeping on writing down. I'm sleeping on writing down.
A lot of things. All right. So the interesting thing about enzymes is enzymes don't typically only use one of these mechanisms. They use multiple, and sometimes all of them, right? So we can't change space, we can't change pressure, we can't change temperature. So we're going to have to change how we interact, how that enzyme interacts with our substrates. And what is that dependent upon? What is an enzyme?
An enzyme is a protein that acts like a catalyst, speeding up a reaction.
It's a protein. So how do we get the sequence of protein? From where?
From the chart.
From, well, and the codon chart relates amino acid sequence to gene sequence. So how our enzymes function is dependent upon natural selection, right? If the DNA changes, then it's going to change how the enzyme functions. And so it's really dependent upon natural selection, right? To optimize that structure-function relationship between them. So if we really want to understand how enzymes function, we're going to have to kind of generally describe our properties of our enzymes. And I don't know why this little thing moved to the bottom. Okay. All right, so here's kind of a standard enzyme. And this is the induced fit model. How's the induced fit model different than the lock and key?
Induced fit model, the induced fit model, the enzyme makes minor adjustments to the shape in order to accommodate the substrate. So
it's in one conformation unbound, it binds to the substrate, and it induces a different conformation of the enzyme. And then once the reaction is complete, it reverts back to that original conformation. So sometimes it's a large change, sometimes it's a small change. It just depends, right? So the enzyme's active site, so right here, this active site right here, is going to provide optimal chemical, optimal physical environments. So optimal chemical and physical environments for this reaction to occur, right? So how could you do that? How can you provide an optimal chemical and physical environment?
Yeah. By making sure all the charges are in
well, charges are important. So look at our substrates. Our substrates have charges, and so do the active sites on the enzyme. And I need to move my chat over so I can see everything, all right? Okay, so what you're going to see is that our substrates fit into our enzyme in a particular orientation. So the enzyme is going to orient the substrate for optimal configuration, right? Because if you look at the top up here, are these two substrates in the correct position for a reaction to occur? No, they're not. But what happens when they bind inside of the enzyme? Now, because of how those positive charges and negative charges line up, we know that they're going to be in the right orientation. And so the interactions between the molecules are going to be productive.
So in regards to their charge and shape.
Charge and shape. Hydrophobic and hydrophilic. It can be more than just charge, but it's all about orientation.
So the charges of the inside orient the substrates and the content.
The charges inside, so everything we're talking about is the active site. The active site of the enzyme, right? Provides the optimal chemical and physical environment. How does it do that? Through orientation of the substrates. Now look at that local environment inside of that active site. You're going to have a very high local concentration of our substrates, right? So you're not actually changing the overall concentration of the reactant and substrate, but locally, you're going to have a very high concentration because you're going to have both substrates present. So you're going to increase local concentration of substrate, of both substrates, or whichever substrates. There's one more way. It's not just that the substrates fit into the pocket. The substrates actually interact with the enzyme. How do substrates actually interact with enzymes? Charge can be one of them, but what do you mean by charge?
Like positive and negative.
What's positive and what's negative? The whole protein? No. What's positive and what's negative? The functional groups, our functional groups. So our amino acid functional groups. These guys are going to participate in reactions with the substrate. So the functional groups can participate in chemistry of the reaction, right? Whatever that chemistry is. Maybe it's positive and negative, maybe it's something else, and we'll talk about what are those things will be. But they actually participate in the chemistry of the reaction. And it's no chemistry you haven't seen before. It's all chemistry you've seen before. So these functional groups, right? These functional groups, they're going to align inside of the pocket. So inside here, inside this active site. But here's the trick. You don't want them to bind too tightly, right? So they're going to bind, so substrate binds due to affinity for, you call reactive side chains, reactive side chains. But do you want that to be really, really high affinity? No, not really. Why not? You don't want it to stick around. You want to change to the product, and then you want to kick out that product because you want to go again, right? It's an enzyme. You want it to be recycled. So you want some affinity, but you don't want it to be too high.
So you don't want it to stick around.
Right. So there's an affinity for substrates, there's an affinity for intermediates, and there's an affinity for products. Where do you think the affinity is going to be the highest? If you have a high affinity for the products, you'll convert to products and then you won't kick the product out. The intermediates, because you want to form those high energy, relatively high energy intermediates so that they move on to products, right? So the highest affinity is between enzyme and intermediates. Okay, so when we look at our enzymes, what we're going to see are there are 3 different physical and chemical properties of active sites that contribute to our catalytic properties. The first one, sequestered microenvironment of the active site. So this one contributes to do you remember delta G dagger? Did you watch the video?
Yes.
What is delta G dagger? Really?
Measure of like
Activation energy. It's really activation energy, right? That's transition state energy. Okay, so this is what we're going to affect when we're making this sequestered environment. So we're providing an optimal orientation of our substrate relative to the reactive chemical groups that are basically surrounding it, right? So this is independent of catalytic activity. So these two are independent of catalytic activity. And we're going to talk about catalytic activity. It's 2 different things, right? So we talked about providing that optimal orientation. It's got positives by negatives, negatives by positives, hydrophobic and hydrophilic, residues are in the right orientation, that kind of stuff. But then what you're also doing is you can also exclude excess solvent. So what's our usual solvent? Y'all? Yeah, water, thank you. Water. So a lot of our proteins are going to form hydrogen bonds with water, right?
So both these are independent of metabolic activity.
So our proteins, our enzymes tend to form hydrogen bonds with water, right? Well, inside of an active site, do you want those residues that are supposed to participate in these chemical reactions to be hydrogen bonding with the water? Or do you want them to be interacting with the substrate? With the substrate, right? So a lot of times what you'll see is these active sites are kind of deep inside of the enzyme. And so that prevents water from going in, right? And so you can make sure that your substrate goes in and your water does not. So they call that a hydrophobic channel.
Hydrophobic channels are
Just like a channel that your substrate has to go through that water won't go through.
good.
And the other thing is that you can have that conformational change. Remember when we were talking about that induced fit, right? So if you're open to picking up a substrate, once you pick up the substrate, you kind of close in around that substrate and you block out the water from entering. So there are a couple of different ways that you can exclude excess solvent. Hydrophobic, the hydrophobic channel, or you can have conformational changes. Right, those are the 2 ways we can exclude solvent.
And the conformational change is just
Like the induced fit. Mhm. Yep. in the enzyme. Conformational changes in the enzyme, not the substrate in the enzyme. Okay, so then we have two different ways that are catalytic, right? So the second one is when we have binding interactions between the substrate and the enzyme to make that transition state. So that's catalytic. And then presence of functional groups, again, catalytic. So we kind of talked about one already. Let's go and talk about 2 and 3. Y'all ready? I feel like it's harder to pause this. Oh, is this a good example of a lipid channel? Yes. Yes, it will keep out.
Channels.
Mm-hmm. All right, so, right, we said the second way, over here, the second way, we're gonna start on the second one, right, is to contribute to the catalytic activity, right? So we're gonna talk about binding between the substrate and the enzyme to make that transition state. So here's our original in red, uncatalyzed reaction. Boy, that was a really big hill to have to climb to get all the way down here, right? That's a lot of energy that had to go in. So our delta G dagger of our uncatalyzed reaction is very high. Now look what happens. We have a catalyzed reaction. But what we're going to undergo is a couple of intermediates, not just one. We're going to go through a couple. Yep, and this high energy, this is very, very, very unstable. Yes. And what I want you to notice is when you're comparing these activation energies, how does that affect the overall delta G of our reaction? What? It doesn't. It doesn't. That's really, really important to remember. It does not change delta G of our reaction. This is exactly the same whether we are catalyzed or uncatalyzed. The dagger, the double dagger, is the delta G, the activation energy. I don't know, I really don't like it written like this.
Yeah, why can't activation energy just have its own?
It does. It does. EA. It does. So this one is EA of uncatalyzed, right? And this one is catalyzed. I think it just gets confusing, but okay. All right, so let me see if we can write out kind of what happens in this reaction. So delta G is a state function. The delta G is the change from reactant to product. So you don't take into account any difference other than where you start, and this is where you start and where you end. So it doesn't matter, like, it's an independent pathway. It's an independent pathway. Like if you wanted to go to Starbucks, right? How many different ways could you take to get to Starbucks? Quite a few different ways. But your change in distance from starting point to final point is the same. And that's a state function, right? It doesn't matter what pathway. Like you could spend a lot of gas, a lot of energy to get from here to Starbucks. But you could also go a different path and consume less energy, but you would still get there. We would want the fastest, I agree, Hannah. I would want the fastest path to Starbucks as well. So does yourself, so do yourselves.
Same way, same thing with getting to the back.
Same thing. All right, so if we were gonna write our standard reaction, right? We have our enzyme, and we're gonna add our substrate to that enzyme. Instead of going straight to enzyme and product, we need to kind of write out what's gonna happen. So what are we gonna form first? Some kind of an intermediate. So that intermediate, that very first intermediate, is it going to be our enzyme that is somehow attached to our substrate. So this is an enzyme substrate complex. And really, I drew this wrong. Really, this should be a double-headed arrow. So going through multiple, yes, Lindsay, going through multiple transition states is way more efficient, And we're going to show you some examples, and you'll believe me in a few days, I promise. Okay, so you have this enzyme substrate complex, but it's still in the form of the substrate. What are we trying to get to? We're trying to get to product. So it's efficient to still be attached to the enzyme, but to make the product. So we call that the enzyme product complex. So this is the enzyme product complex. And then what are you going to do with that product? We're going to release it. And we want our enzyme to look exactly like it did when we started the reaction, right? So we're going to end up with enzyme plus product. So let's talk a little bit more about this. Let me go down, but don't. I want to make it bigger. Can I do that? Okay, hang on. Y'all, it's me trying to figure out technology. I want it up here. All right. So if we look at our first transition, look at your first transition here. When we go from enzyme plus substrate to our enzyme substrate, this is going to require energy. All of these steps require energy. Where do they come from? Why energy comes from? Where? Any ideas? No, it actually does not. It actually does not. So I know, right? Not you consume energy when you break bonds. You're going to consume, you always consume energy when you break bonds. So we have to be making something, right? Well, bonds are a lot of energy to make, right? So what's something that requires less energy that we can make? Weak interactions. What are weak interactions? Hydrogen bonds, but they're not really bonds, which is why I hate that they are called bonds, FYI, in case you didn't know that about me. Ionic interactions. What else? Van der Waals interactions, right? These are all things that we've talked about before, right? When we talked about folding of proteins. So weak interactions between what? Between the enzyme and substrate. Yes, ionic, van der Waals, hydrogen bonding, the hydrophobic effect, right? All that kind of stuff. Okay, we're still in the form of the substrate. When we do our next transition, when we go from enzyme substrate to enzyme product, now what are we doing with some bonds? This requires what? Breaking, now y'all are on the right track, breaking, making bonds. Right? You have to break and make bonds.
Is still the same hydrogen, ionic, and van der Waals.
No, now we're making covalent bonds. Now we have to make covalent bonds. That's why I hate hydrogen bonds being called bonds, because they're not really bonds. So you need the active site residues. Active site residues. These residues are going to facilitate contacts, right, with our transition molecules. These are our two transition molecules. You got 1, 2 transition molecules. To make them stable, to allow those bonds to form. So you actually get a gain of energy here. Where's my mouse? Gain of energy from formation of bonds between enzyme and substrate. Well, enzyme and product substrate, we'll put it that way. Because how do you get energy? You can't get energy by breaking a bond. You must get energy by forming a bond.
A bond between.
Enzyme and product / substrate. Come on, eraser. Okay, now tell me about the end. What happens at the end? I'm running out of room. How about here when we're going from our enzyme product complex to our enzyme product? Why is this going to be released? Why is the product released? Where can I write this? Product is released. Yes, you do break some bonds, but why do you kick that product out? What did we talk about just a few minutes ago as to why we kick out the product? Where do we have the highest affinity for the enzyme and all this stuff that's going on?
Intermediate.
The intermediate. So when you transition from the intermediate to the product, the enzyme is no longer basically attracted to it because of reduced affinity of enzyme to product. It has a higher affinity to that transition molecule. So it's preferred to kick that product out, pick up a new substrate, make a new transition molecule. Yeah, because of low affinity. Good, low affinity. Y'all got this? Y'all ready? I feel like, I don't know. Yeah? All right, so how do we know this is true? We can support these theories with actual data. And if you can't support it with data, it's not real science. So always, always, always check to make sure it's supported by data. So we can use what are called transition state analogs. These analogs, what's an analog? Anybody know the definition? Not the definition? An analog is something that's very, very similar to a substrate. So if you have an enzyme that works on a particular substrate, if you can make a synthetic peptide that mimics your substrate, we call that an analog. So it's a molecule that mimics the conformation, the side chains that are important in the reaction.
Where is it?
But you have this slide, Rolf.
I know.
Okay, you have the slides. All right, so you have this enzyme. This enzyme is called adenosine deaminase. It takes adenosine and it deaminates it, imagine that, into inosine, right? Don't you love how they name enzymes? Isn't this like the most magical thing in biochemistry, that they're actually named properly? So what we think happens is that we turn into a transition conformation. So I want you to look right here at this carbon on our molecules. When it starts out, how many different areas of electron density do we have? Like back to Chem 101, this carbon right here has 1, 2, 3 areas of electron density, right? A double bond counts as one area, a triple bond counts as one area, a lone pair counts as one area. Yes? Okay. So what happens when we transition from our substrate into our transition molecule? Now we have how many? 4. What's the term for 4? Tetrahedral. So it would be tetrahedral. So we think that our intermediate makes a tetrahedral conformation before we then make the product. So what we can do is we can synthesize this analog, and this analog is going to mimic that tetrahedral conformation. So is it a perfect match? No, right? We have a hydrogen here. Here we have an NH2. It's not quite the same, and I'm telling you that this R group is not quite the same. But this is what we think is really important in the interaction. And so when we use this analog and then we look at the binding affinity, we know that our enzyme has a binding affinity of 10 to the negative 5. So this is our enzyme to substrate, right? If we put in this analog has a 10 to the negative 13 affinity. Is that tighter or weaker affinity when we go this way? It's the opposite. It's the opposite. So this is a stronger affinity. So remember, this is like our dissociation constant, right? So this is how much will disassociate. So when you think of affinity, you have to swap it. Remember that? Okay. Right, the KD, right. Okay. So when we use this analog, oh my God, how much more affinity is that? That's huge. This is 10 to the negative 5 versus 10 to the negative 13. So this is really cool though, because what we can do is we can actually inhibit enzymes with this methodology, right? We can construct peptides to inhibit enzymatic activity. That could be really, really important for a pharmacological drug, right?
So what you're saying is, like, if the charge, the affinity is too high, you can actually inhibit them.
Correct, correct. So what we really wanted to know was like a structure. Can we put something in the binding pocket that mimics our substrate and then get a structure from it? Yes. But the off-topic thing that we can do with that is then we can use this to construct an inhibitor. And we'll talk about that in another chapter later. But anyway, okay. So that was like a sidetrack. All right. So we know that these transition conformations are super important for enzymatic activity. And that's what this information is telling us, that yes, those transition molecules bind very tightly and are very, very low energy in terms of making a reaction go. Okay. All right, so the next way, what was the number 2 was presence of catalytic functional groups. Remember, if we go back here, number 3, presence of catalytic functional groups. So this is what we're going to talk about next. Or were we here? Presence of catalytic functional groups, right? So we know our functional groups, right, are sitting in a pocket. They're away from the solvent. And so you're going to have these nearby reactive chemical groups on our amino acids of the enzyme or on, so these could be on the enzyme, or what else could they be on? What helps enzymes work? Yes, cofactors, coenzymes, cofactors, however you want to think about that. It's the same thing. Coenzymes, cofactors are the same thing. They're 2 different words for the same thing. So these functional groups are going to interact with our substrate. And they're going to have one of 3 different kinds of reactions happen. You can have acid-base interactions. When you have acid-base interactions, or we call that acid-base catalysis, because it's an enzyme, right? And enzymes catalyze things. What are you going to be doing? What do we do when we talk about acid-base reactions? I know that's your favorite topic. Acid-base. What happens in acid-base reactions? What gets moved around?
Electron, hydrogen atoms.
Protons, hydrogen atoms. Protons get transferred. Proton, we'll just do proton transfer. So protons can transfer from different substances, right? We know that we can get a proton off of water, right? Water can donate a proton. So if it's water, if it's from water, we call that a specific acid-base catalysis. If you get it from one of these functional groups that are in the list, functional group, we call that general. Because there's not one specific functional group that donates or receives the proton. It can be a whole group of them. But with water, it's always going to be from water, and so we call it specific. So look at our groups over here. If you're in the general acid form, what are you going to do? You're going to donate or you're going to receive? You're going to donate. Base form, you're going to receive. And anybody remember the special thing about His as a functional group? It has a pKa close to pH 7. So it can act as both depending on the local environment, right? So you're going to see His a lot. You're going to see His a lot. Is it? I don't know that I have numbers on it, but you are going to see it a lot. All right. So you can have acid-base. So this is one way that you can do it. You can have acid-base transfer. And so we're looking at all of our functional groups here. You could also have covalent catalysis, where you actually make covalent bonds with the substrate. And then you can have metal ion catalysis. And so we're going to talk about each one of them. So let's show an example of acid-base first. All right? No, yes, no. All right, so here's acid-base catalysis. We're having that proton transfer. And we said that proton can transfer, can involve water, we call that specific, or a functional group, and that's general. Okay, now I really want to be able to zoom in because it's hard to see this stuff. More options? No. Oh. No, I don't know how to do that. I'm gonna have to figure that out. Nope. Oh, y'all, so sad. I love doing this on my iPad, but I can't do that. That's okay. All right. So this is the example that we're going to talk about for acid base. So this is a pancreatic ribonuclease, and this is going to degrade RNA. So that's what this enzyme does. Is the whole point is to degrade RNA. So here's your RNA molecule. So your RNA molecule has a sugar phosphate backbone, right? And we're just showing the base here just as a base because it doesn't really matter which one it is. Because this enzyme is going to cleave the backbone. So how does it cleave that backbone? It participates in acid-base reactions. So look what kind of residues we have participating. Look at this, a His. What's over here? Another His. Y'all have these notes, right? I know y'all are straining to see the screen, but y'all have these notes, right? What? Oh yeah, it's the 2 histidine residues, right? Okay, let me erase my other arrows. Okay, so what's gonna happen in here is that our hist is gonna act like a general base. So this is gonna act like a base. So what's gonna happen in a base? It's going to accept a proton. So here's your hydrogen that's going to be accepted by the nitrogen on our ring. What happens when this hist pulls a hydrogen off? Now what do you have? You have this oxygen that's going to have a negative charge, right? So it's going to undergo a nucleophilic attack on our phosphate group. So then what's going to happen to that? Oh, we have another hist down here. This hist has a hydrogen that it can donate. So what's going to happen is we're going to break the bond between these 2 RNA molecules, RNA bases, right? So look at what we cleave. We cleave right here, this phosphorus and oxygen bond right here. And so see what happens? Now when that happens, we release one base. One residue. This requires water. Now our hist down here, right, can act as a general base. The hist down here acts as a general base. It's going to look at the water, now that you have room for the water to come in, and it's going to pull a hydrogen from the water, and what's that going to leave? A hydroxyl group. That hydroxyl group is going to attach to our phosphorus here. So do we have an intermediate form? Yes, we have an intermediate form. And does it associate with the amino acid residues in the enzyme? Absolutely, right? So this is one example of acid-base catalysis where our residues can act as donors and acceptors of protons in order to facilitate making a transition molecule before we make a product. So tell me about the orientation of our, and the configuration of our side chains within the enzyme from when we start to where we end. How does it look? So here's where we're starting, and here's where we're ending.
So it cleaves off, so the full page cleaves off one of the.
It cleaves off one residue. So what's going to happen is at the end, now our enzyme looks exactly like it did in the beginning. And our RNA molecule will slide down one and we'll go again. And we'll cleave the next residue off, right? And it just continues and continues to cycle. All right, let's look at, so that's acid base. The next one we need to look at is our covalent catalysis. Covalent catalysis occurs not for acid bases, but these are nucleophilic attacks. So we have a nucleophile. What's the definition of a nucleophile? Any takers?
It's when they desire a nucleus.
This one's electron rich. Yeah, this is the attacker. This is the attacker. Electron loving, electron rich, right? It attacks an electrophile. What's an electrophile? Electron deficient, electron poor, right? And because of that attack, now we're going to form a covalent enzyme substrate intermediate, right? So here's our example. We have our substrate, right? We have a side chain. In this case, what is NAD an example of? A coenzyme, a cofactor, okay? All right, so what's going to happen here is we're going to actually form a covalent interaction. So where's our covalent interaction happening? Between our sulfur and the carbon on the substrate, right? So we're gonna have an intermediate here, and we actually have two intermediates. So here's our first intermediate where we have a tetrahedral configuration, but then our NAD plus, look what it's gonna do. What's it pulling? It's gonna pull your proton. It's gonna pull your proton off, and now do we still have that tetrahedral configuration? No, right? So we change the conformation. We're gonna kick out NADH. We're gonna use another NAD molecule, and we're gonna covalently attach a phosphate group. So we have a nucleophilic attack by our inorganic phosphate group to form an acetyl phosphate product. So here's our new product that has the new linkage between our substrate and our inorganic phosphate, right? So this part of the molecule has the original conformation, but it had to go through an intermediate in order to get to that final step. So it kind of feels like you're going through too many steps, but they're actually low energy steps, and it's beneficial. It speeds up the reaction.
Is it always covalently attaching inorganic phosphate?
No, this is just an example. It could be many different ones, but attaching a phosphoryl group is super common in a lot of different reactions. Okay, the last one is our metal ion. So when we have metal ion catalysis, our metals do a couple of different things. One of them is to promote proper orientation. If you have a metal ion, what's the charge on your metal ion? It's always going to be positive. Always going to be positive. So it's probably going to interact with some negative portion of the substrate or the intermediate, right? And so that's exactly what we see, but it can also aid in redox reactions, which we can talk about. But here's, for an example, an enzyme that has zinc, right? And so the zinc is actually going to promote a formation of a hydroxide anion that's going to act like a nucleophile. So the nucleophile is going to attack the carbon. Now we're forming this intermediate, right? Then our zinc can interact with water, right? And it can create a new product, and then we can release this. In this case, we're starting with carbon dioxide and we're going to end with bicarbonate. So we needed to add a hydroxyl group, right? So we had this initially from water, then we can attach it to our carbon, and we'll release a proton in the meantime. But we can do that with our metal ion. All right, and we're almost at the end. How much time we got? 2 minutes. 2 minutes. 3 minutes. Oh, I like that. All right. So when we talk about these ions, we can call them coenzymes. And so when we talk about coenzyme-dependent redox reactions, you'll see a lot of them NAD plus and NADH. I know you've heard that before. Sometimes FAD, FADH too, right? These are ones that are involved in a redox. So anytime you hear about a dehydrogenase, that's going to be a redox reaction. So we're going to look at this a lot. All right. One example is when we convert from pyruvate to lactate using lactate dehydrogenase. So the pyruvate itself is reduced, right? And you know, in an oxidation reduction, one species is reduced while the other species is oxidized. So we remove those hydrogens and we're going to attach them to our pyruvate to make lactate. So sometimes these coenzymes, these cofactors, really are there just for oxidation reduction purposes. All right. You can have metabolic transformation reactions. So a lot of times you don't actually change the molecular formula in a reaction. All you do is change the orientation of the atoms and what they're attached to. So we call that an isomerization. Y'all studied those before in organic, right? Yeah.
I think
Condensation reactions. How do you know it's a condensation reaction? What are you making?
water.
You make water, and you usually combine two substrates to make one, right? A dehydration or a hydrolysis is the release of water, so it's the opposite. So it's the opposite, right? So this one is an isomerization, where your number of your different atoms have not changed. You're just putting them in a different orientation. This one is a condensation reaction right here, where we're taking two things and we're making one longer molecule here. So a lot of times in these transformation reactions, these are like the anabolic and catabolic. Anabolic, catabolic, K-T-A-B, catabolic reactions, right? Those are those kinds of pathways. And then our very last one for today is reversible covalent modifications. So you'll see these all the time. Something gets phosphorylated, and that either activates it or deactivates it. And it's reversible because you can take that group off. So remember when we talked about kinases and phosphorylases? Kinases do what? Add the phosphate, phosphatases take it off. So where do you get that phosphate group from? ATP. Yeah, and that's how you regenerate, right? So it doesn't have to be a phosphate group. It can be other things, but that is the #1 most common in all the cells. So I think I'm done. I think that's the time anyway. So questions? Hannah, you want to talk? Yeah, you can talk. Just hang tight. Everybody else good? So you have the lecture for next time to watch. It's only one lecture, and it's already posted, 7.3. Yeah. It's too much going on in my brain. Once we hear something about what happens with the hurricane, I'll send y'all an email. But be on the lookout, watch your emails. Y'all have a good rest of your day. All right. Have a good afternoon. Y'all good?
I might have to try to use this in the session because I need to go to the hospital at 10 a.m. to get a heart monitor hooked up.
Oh yeah. It's all recorded. It's all going to be recorded, so you know how to get to the YouTube channel, and you're good.
All right.
You're good. No problem.
I have to go do something quick, but I'll say next. I expect a few questions. If I read email you a few of the questions.
Sure.
I worked on those other questions.
Awesome.
Okay, just expect a few questions asking for a little bit of help later on in the day.
Sounds good. I can handle that. All right. Let's see who's still on. Anybody on have a question? I have to be to guess. It's fine. Madison, you're good. You're good. I understand stuff like that happens. Don't worry about it. You're good. All right. I know Hannah has a question, so if anybody else has anything, and then I'll let y'all get off and Hannah and I can chat. Okay, I'm going to stop the recording. I'm going to try and stop the recording. There we go.